Right, we're going to do plant transport now to cap off module 3 for OCR. So let's just get into it. Again, we always try to begin, oh that's not very good, with why. Why do we need the transport system in plants? Okay, so the same issues around the surface area, the idea that the plant is a large, relatively large, or plants can be very large multicellular organisms, and because of that the surface area to volume ratio is comparatively quite low. You can argue some parts of the plant have are designed to have a high surface area to volume ratio, for example leaves, maybe roots, but if collectively overall including the stems and everything else, there's a relatively low surface area to volume ratio, so we can't rely on diffusion. Second, we do have very specialized parts of the plant, so you have roots. So the roots are absorbing water, they're absorbing mineral ions, but the leaves can't do that. In contrast, the leaves, they're actively, they're photosynthesizing, so they're producing lots of assimilates, amino acids, sugars, things like that, that can help the growth of the plant. But that's being done at the leaves, not so much at the roots or the stem. So for this reason, because you've got these specialized parts of the plant that are doing different things, absorbing or producing different things, we have to have a way to get them to the other parts of the plant. So the water and the mineral ions that the roots absorb need to be transported to other parts of the plant. The sugars, other organic molecules produced by the leaves, need to be transported to other parts of the plant. And so we need that transport system to do that. Actually, we'll go xylem and phloem first as the tissues, and then we'll look at how they might be distributed in different organs in the plant. Okay, so let's go to xylem structure. So xylem, and here we have phloem. Okay, let's just have quick pointers, key points about their structure. Xylem, non-living cells, right? So they're cells or xylem tissue is formed of cells that used to be living, but it has lost all its living contents. The cell wall has been impregnated with lignin, so cell walls have been lignified. Okay, and that's kind of a waterproof, very high tensile strength substance, which has resulted in the living contents of the cells being lost. Their end plates have been lost, so the cells join end to end. Okay, just indicate that here. The cells are joined end to end to form continuous hollow tubes. Okay, so that's the lumen there. Right, we've got the wall here, xylem wall, right there. Got the lumen, which is this space in the middle. We have these pits, which allow lateral movement. So not only can the water move up and down the plant, but it can move across the plant too. Okay, so there we have that. And then we move on to the phloem. Now the phloem is living tissue. It is living, however, it still manages to perform a transport function. Now, it's living, but you can kind of think of it that it's on life support kind of thing. Two main types of cell make up the phloem. First, actually the first, is these sieve tube elements, okay? And they have close by these companion cells, okay? Now they are connected to each other by these plasmodesmata, Okay, so stuff can be exchanged between the two cells. The sieve tube element, you will have noticed, it doesn't contain any of the organelles, right? So there's no nucleus, endoplasmic reticulum, Golgi apparatus, any of that. All of those functionalities are provided by the companion cell so that the sieve tube element doesn't need those organelles, and so there can be a kind of more unobstructed movement of stuff. Okay, the fluid in the sieve tube elements can therefore move unobstructed, which aids in the mass flow of the fluid in there, and thus all the stuff in there can be transported, and we'll go into more detail of that in a bit. Okay, but those are the key structural elements. Let's just label. So these are the end plates, perforated end plates. Again, they're perforated, which allows the movement of the fluid, and it's not giant things that are moving through this stuff, so the perforations will do. What else? So this, you can call that a cell wall. You might not be able to call that a cell wall here because these aren't really cells. Right, and the plasmodesmata, right? So I don't like to go across. I'll just draw a few here maybe. Okay, plasmodesma. All right, so that's kind of structurally the xylem and phloem, and we'll come back to how those structures feed in to or connect to their functions. Okay, next let's see how these tissues are distributed in different organs of the plant. Okay, so let's label these diagrams up and then just quickly look at where we would find the xylem and the phloem. All right, so first up here we have a stem, then we've got a root, and finally we've got leaf. Okay, so these are different organs of the plant. They do different things, right? Stem has primarily a support and transport function. The root has primarily a absorbing water and mineral ions function, and leaves are primarily there for photosynthesis. So again, organs adapted to perform particular functions, but they all have xylem and phloem. So on the outer part of the stem, you do have the epidermis. The next layer in is the cortex, so that will be right here. Okay, then you have this vascular bundle, which we'll come back to. So this structure here, that's called the vascular bundle. And by vascular, that's what we're referring to, xylem, phloem, okay, collectively. So that whole group of tissues there, that's called vascular bundle. And then right on the inside, we have the parenchyma. Okay, now, it is the vascular bundle that contains the xylem and the phloem. So the xylem is on the inside, so you might need to remind yourself of this. The xylem is on the inside part of the bundle. The phloem is on the outside part of the bundle, so that's right there. Okay, and then in between the two, you have the cambium. Okay, right, it's not arrows, but cambium, which contains or which has the stem cells that can divide to form phloem and xylem as per requirements. Okay, so there we have that. Root. Now, the root has got an extra layer, so it's got this exodermis, which contains the root hair cells. Then we have the epidermis, so the next layer in. Next layer in, so that's this one right here, we have cortex. And then we have the endodermis. And then we have the xylem then is this cross-shaped tissue. So that's our xylem. Okay, that's the xylem. And then between the arms of the cross, we have the phloem tissue. Okay, and that is the root. Next, we have leaf. So just for navigational purposes, this right here, that would be the palisade, photosynthesizing cells, right? And then we have here, so it's at the top that we have the xylem. This is our vascular bundle, okay? And at the bottom we have the phloem. Okay, and there we have the distributions of the tissues, and that's going to take some work to make sure that you know what that is. But a number of practices sketching this out, you can see how kind of quite simple it is really. But a number of practice goes at sketching these out, labeling them should do the trick. All right, let's get into functions now. The first thing we're going to do is look at water and how it enters the root. Then we're going to see how it gets into the xylem, and then we're going to follow up with what happens with how the water moves through the plant then towards the leaves, and how the transpiration stream is involved in the movement of water through the plant. The roots, the root, you can think of it as you can think of the root as an exchange organ of sorts, okay? It needs to gain something from the environment, and its adaptations are to maximize the rate of those processes, okay? So we've got a high surface area, we've got minimized diffusion distances, okay? And we've got the existence of concentration gradients. So as we've discussed before, those three things help to maximize the rate of this passive process, okay? Now, in the exchange organs that we've discussed before, that was diffusion, but in this case, because we're talking about water and essentially the diffusion of water, we are referring to osmosis, right? So, water movement of water into root, okay? And we're going to follow the journey about how that water is then going to get into the xylem from the root, okay? Now, it's quite straightforward really. You know, the root is, or the root cells, they are cells. And their cytoplasm is going to have a lot of stuff dissolved in them. Because they're cells, they're going to have sugars in order to survive and carry out respiration. There's going to be amino acids so that they can continue to build their proteins, etc. There's going to be mineral ions so that the cell can maintain its ionic concentrations and balances, pH, etc. So there's going to be a lot of stuff dissolved in the cytoplasm of the plant cell, and for that reason, it is said to have a low water potential. Now that's not an arbitrary thing; it's a relative thing. It's got a lower water potential than the water or than the fluid that is out there. Okay, or the solution. So you're going to have some soil particles, right? And around those soil particles, you're going to have some water. And yeah, that water is going to have some stuff dissolved in it as well. But compared to the cytoplasm of the root hair cell, it's going to have a much higher water potential. Okay, so there's a lower water potential here. Effectively, there's a higher water potential out here. And because of that, what happens next is pretty straightforward. If my pen would work properly. Okay, so water is going to move by osmosis passively into the root hair cell, and thus we have movement of water from the area surrounding the root into the root itself, simply because the root hair cell has a lower water potential. Okay, what's going to happen next is that water needs to move into the xylem so that the rest of the plant can benefit from the gain of water that's happened at the root. All right, so what we've said is because water has moved by osmosis from outside or around the root hair into the root hair by osmosis. Okay, what happens next? Well, water, because now the water has entered the root hair cell, now compared to the other cells around the root hair cells, okay, where are we talking about here? We're talking in the exodermis and epidermis, right? Now, the cells in that area, they are going to have a much lower water potential compared now to the root hair cell, because all this water has entered the root hair cells, right? All this water has entered the root hair cell. So that makes the water potential in the root hair cell much higher than the neighboring cells. And so water is going to move down the water potential gradient from the root hair cells into the neighboring cells in the exo and epidermis. Okay? And so it will continue all the way into the xylem. Okay? All the way into the xylem. All right. Now, we do need to be aware of the pathways that water can move. So remember, there's those two terms, right? Yes, the general principle is osmosis. Okay? The gain of water into these cells and the kind of chain or the and then the chain of events that causes, right? Because of the established water potential gradients, water is always going to be moving into the less concentrated cell. Sorry, into the next more concentrated cell, or the next cell that has the lower water potential. Okay, that's the general principle, is osmosis. Okay, but then there's two paths that the water can take, right? So when the water goes from the cytoplasm of one cell to the cytoplasm of the other cell, remember that plant cells have that these gaps between them, right? That the water can move from cytoplasm to cytoplasm, right? So from cytoplasm to cytoplasm, that is called the symplast route. Cytoplasm to cytoplasm, so that's symplast. Okay, and I think that's what we are familiar with. But there is also this other route that the water can take, because the cell wall is permeable to water, and so the cell wall is also a route that the water can travel through. So I'm just going to highlight that in red. My red pen's not going to let me down. So the water can also travel through the cell wall. So from cell wall to cell wall. So through the cell wall. Okay, now that route through the cell walls, is called the apoplast route. Apoplast route. Okay. And that's fine. However, when the water gets to the cells just outside the xylem, that water needs to be kind of drawn or kind of directed, guided into the xylem rather than spreading through the tissue of the root, it needs to be directed into the xylem. So in order to direct the water into the xylem, the cells just outside the xylem have got something in their cell walls, I don't know if you remember this, called the Casparian strip. Okay, now this is a waterproof material, black. Okay, this is a waterproof material in the cell walls, kind of like forms a ring around the cell, so within the cell wall, okay? But what that does is it prevents the apoplast movement of water through the cell wall into the neighboring cells, and rather it kind of forces all the water at this point into the symplast root and therefore into the xylem, okay? So at this point, all the movement becomes symplast and it goes into the xylem rather than just kind of spreading out through the tissue. It's directed into the xylem, down that water potential gradient. And thus we get water into the xylem. No, I did forget something actually. Now, how do we, yes, one way that we get water into the xylem and we're not kind of letting it hang around in the cell walls is the Casparian strip. That's one thing. The other thing is that these cells just outside the xylem, they are also actively transporting mineral ions into the xylem, lowering its water potential. And that kind of increases the gradient, increases the water potential gradient so that we maximize the movement of water into the xylem. So that's an important thing to remember, not something that you should forget. Okay, so at this point, the water's in the xylem. What happens next? So next we're going to talk about transpiration, because it is the stuff that happens at the leaf, as well as the stuff that's happening here. They work together to get that water moving up the xylem, because as we're causing water to go into the xylem here, we're building up the pressure. So there's a pressure, there's lots of water in the xylem, and that's creating a pressure, okay? And one of the ways that pressure, that if there's a high pressure here and low pressure in other places, we've discussed this already, that's a pressure gradient. And just like things move down chemical gradients, or they might move down water potential gradients, they can also move from areas of high pressure to areas of low pressure. And that's one of the forces that causes water to move up the xylem vessels to other parts of the plant. Okay? But what is happening at the leaf that might result in there being a low hydrostatic pressure in other parts of the plant? So let's now look there. So this is a leaf here. I've got a xylem vessel. I've got a xylem vessel here, and I've got some cells, and this is the edge, this is the epidermis of the leaf, and that's a stomata. Now, water is being supplied to the leaf, right? So as the water comes out, it's going to move again through the symplast pathway from cell to cell, or again, it could also equally move down the apoplast pathway from cell to cell. But why? It doesn't just move for no reason. Why is it moving? And this is important, guys, that all I'm doing here, you kind of already know the stuff. If you're revising, you've heard this stuff before, you've seen it before, you know it. But what my philosophy is that if you know why all the stuff is there, if you know all the stuff, if you know the concepts that allow this stuff to stick together, then you can remember it and then you can use it better. Anything that helps, any story, any narrative, any explanation that helps you understand why you need to know this, that's going to help, okay? And that's what I'm trying to do. So, why is this water moving? Well, evaporation. So we have these gaps, right? we have these gaps called stomata, and water can be lost through these gaps, and it will be, because outside in the air there's a low water concentration, right? It's air, right? And if there's a low water concentration here, and as a living organism there's loads of water in any living organism, right? So there's going to be there's always a gradient. So if there's lots of water in here and a lot less water out here, we're going to have a gradient. And therefore, what tends to happen is water evaporates. Water evaporates through the air spaces and through the stomata, and water leaves the leaf, okay? And because of that, then, there's always this gradient. So if water is leaving the cells in the leaf, then we could say, well, these leaf cells, they have, because water is continuously leaving the leaf, cells in the leaf have a low water or lower water potential than the xylem, okay? Because the xylem's got lots of water. The leaf cells are constantly losing water, and therefore they have a much lower water potential than the xylem, and that's the reason, that's the driving force for water to be continually leaving the xylem into these cells, right? Apoplast pathway or symplast pathway, doesn't really matter, but yeah, okay? So water evaporates away from the leaf, but that's why it goes from the cytoplasm into these cells by osmosis. Okay? And because water is continually leaving the xylem, it's going to cause an impact on the water in the xylem, right? So because water is being lost from the xylem then, but then we have this concept, we have this idea of water. So what? Water is being lost from the xylem, so what? Well, so this cohesion tension. This is where the cohesion tension theory comes in, right? Because we're losing water from the xylem, but remember, water molecules are cohesive. Water molecules are cohesive, and therefore, when water is moving in this direction, they will tend to pull through their cohesive forces, the hydrogen bonding between water molecules. When one water molecule goes, it's likely to pull others along with it, okay? And because of the cohesion, there's going to be a tension created between the water molecules in the lumen of the xylem vessels, right? And so as the water leaves here, it kind of pulls on the other water molecules further down in the xylem vessels by tension. So the cohesion results in tension, and the tension is what results in the pulling up of other water molecules further down in the xylem tissue as far as the root. Okay? So not only do we have this much lower hydrostatic pressure, which and compared to a high hydrostatic pressure in the root, and fluids will move down that pressure gradient, but as well as that we have the cohesion tension resulting from the loss of water from the leaf. Okay? Right? And that essentially, that's the transpiration stream. Yeah? In totality, in overall. Okay, so that's movement of water through the stem from the roots. Okay, and now let's talk phloem. Okay, so phloem, remember phloem is about the movement of glucose, not glucose, but sucrose and amino acids produced by photosynthesis. Okay? Now, places in the plant that are producing these so-called assimilates, places in the plant that are overall net producing them are called sources. Okay, now a good example of that is an actively photosynthesizing leaf. It's producing lots of sugars, producing lots of amino acids due to photosynthesis. In contrast, in other parts of the plant, they're net using up those molecules because they're not really very good at producing them at this moment in time. Okay, the picture can change. So these other places, oh no, they're not called sources. These other places are called sinks. Okay, these other places are called sinks. So we've got sources versus sinks. Now, again, we're going to be talking about pressure gradients, but what creates the pressure gradient? Okay, so we have sources, they're producing lots of assimilates, sucrose, amino acids, etc. Now what happens is the companion cell actively transports or uses active transport to load up, to actively transport lots of sucrose, lots of amino acids, sucrose, amino acids, sucrose, amino acids. It uses active transport to move lots of these assimilates into the sieve tube element. Okay. What this does is it lowers the water potential at the source. It lowers the water potential in the sieve tube elements, in the phloem, at sources. Okay? And because it's lowered the water potential, and remember, phloem is usually not very far from xylem, and what we've suddenly done is lowered the water potential. So what this is going to do is it's going to invite lots of water to move into this area by osmosis. Okay? And because of that, it is going to increase the hydrostatic pressure. So I hope that chain of events made sense. We made lots of assimilates. Those were actively transported into the sieve tube element. This lowered the water potential, and so water moves in by osmosis. Because suddenly lots of water is coming in here by osmosis, we've increased the hydrostatic pressure. Hold that thought. Now we're going to see the picture at the sink. And I hope you can follow along. It's kind of the same but opposite procedure. Sinks are using up sugars, they're using up amino acids, but not producing them. So overall, we're losing, all right? We're losing amino acids. So amino acids, sucrose, whatever, they're being used up by the sink. What that means is, relatively here, we've got a very high water potential, right? Because we don't have as much solute, because it's constantly being used up and not produced. So we've got a high water potential here. Now, because we've got a high water potential here, water tends to leave by osmosis. Okay, water tends to leave by osmosis, and as a result of all the water loss, we have a much lower hydrostatic pressure. We have a much lower hydrostatic pressure. And so what we have is, in places that are actively photosynthesizing in sources, we have a high hydrostatic pressure. In the sink areas of the plant, where overall assimilates are being used up, we have a much lower hydrostatic pressure. Lo and behold, we've now got a pressure gradient. So as long as we've got a connection pathway through the sieve tube elements of the phloem, as long as these two areas are connected by the phloem, we're going to get movement of fluid from the high pressure to the low pressure down the pressure gradient, right? This is called mass flow. It's basically how blood moves through blood vessels, you know. But when blood moves through blood vessels, the pressure gradient is created by the heart contracting, right? But in a plant, there's no heart, but there's still this existence of the pressure gradient, but the pressure was due to the presence of solutes in one place and the absence of solutes in another place. Okay, so that's translocation, and I hope you see how that makes sense. You know, places, therefore, assimilates are always moving from places where there's lots of them to places where there's less of them. And that's a fair system. Coming back to water movement though, and the last bit is going to be about water, okay? And the first thing we're going to look at is how to measure transpiration with a view to looking at factors affecting transpiration, okay? Now the device that we use to measure factors affecting transpiration, and remember transpiration is really to do with the water movement through the plant, is a potometer, potometer. And I'm going to apologize if you were expecting something complicated, but it is nothing more than basically a plant shoved into a tube. It's important to have an airtight seal, okay? And these tubes are filled with water, right? And there is another tube here where we can adjust, but here, this is important here, we have a bubble, okay? And that bubble is going to allow us to measure or visualize or observe the movement of water through the tube, okay? So as water evaporates away from or out of the leaf, remember what that's going to cause? It's going to cause water to move into the leaf from the xylem, and therefore it's going to cause water to be drawn up through the stem or through the xylem vessels in the stem. Okay, and as that water gets drawn up, cohesion, tension, forces apply, and the water in the tubes is pulled up as well, right? So the movement of the bubble, any movement of the bubble should be proportional to the rate of transpiration that is happening from the plant, okay? And that's basically all there is to it, right? So if there's if the rate of transpiration is high, if the rate of water loss from the plant is high, then the bubble will move more in a given amount of time. Don't forget your stop clock, okay? If you want to measure rate, you need not only the movement of the bubble, but the time in which it occurred, right, okay? So you're going to have a distance moved by the bubble, which will equate to some kind of volume of water, right? So you will need to know the dimensions of the tube, radius, etc. So once you figure out the distance that the bubble moves, once you know what volume of water that corresponds to, and you know the time that it took in order for that amount of water to be lost or to have transpired, that will give you a rate of transpiration. And then what you can do is you can set this experiment up in different conditions to see how those different conditions affect the rate of transpiration. Okay, so what are those factors? Let's just go through the factors and just make sure that you understand the reason why those factors affect the rate of transpiration. Light, humidity, temperature, air movement, and soil, water supply. Light. Now, when there's lots of light availability, that increases photosynthesis. Photosynthesis requires CO2, and the plant would need the oxygen to escape, right? In order to allow access to the carbon dioxide and to allow a route of removal for oxygen, we need the stomata open. And when the stomata opens, that is going to result in more water being allowed to evaporate, so that should increase the rate of transpiration. Humidity. So humidity is essentially the water concentration in the air surrounding the plant. So if the humidity increases, the water concentration increases. If the water concentration out here is higher, that means there's not so much of a gradient for the evaporation of water. So increasing humidity, so increasing light increases transpiration. Increasing humidity should decrease transpiration because we're not getting as much evaporation of water. Temperature. Temperature can have a couple of effects, but I think the most obvious one is that if you increase the temperature, you're going to increase the rate. So increasing temperature increases the kinetic energy of molecules, particles. And so if we're increasing the kinetic energy of water particles, in this case, we're going to increase the rate of evaporation. Okay, so increasing temperature increases evaporation. So essentially, not only should we be able to say it's going to increase transpiration or decrease transpiration, we should be able to link it up with a particular explanatory point, more than one if possible. Okay, so increasing temperature increases evaporation, therefore increases transpiration. Air movement. If you increase air movement, again, that's going to increase evaporation, and that should increase transpiration. Okay, I hope this is making sense. Finally, soil water supply. Okay, now if you decrease the soil water supply, you might not be able to continuously draw up more water. So this might reduce transpiration, because remember, if there's a lack of water supply, then the plant will close the stomata in order to reduce the rate of water loss through the stomata. Okay, so an increase in water supply should increase transpiration, but a decrease in water supply should decrease transpiration, because the plant is going to respond by trying to conserve water by reducing transpiration. Right, adaptations then of plants. So continuing this theme of kind of water in plants, we are going to look at adaptations of xerophytes. Now this is usually adaptations to lack of water, but the adaptation of hydrophytes, that is due to an abundance of water. Okay, so let's have a look at what those adaptations are. Again, same idea as this. We want to know the adaptations, but we want to have an explanation of why that adaptation, or what's the biological reasoning behind that adaptation. Okay, I might run out of space here, but again, if I just got a few down here just to illustrate the point, and then maybe you guys can fill it out, or kind of, flesh it out a little bit more if required. But what we have, waxy cuticle. Right, now the waxy cuticle, that is going to reduce the evaporation. So that's the explanation. So this is all about water conservation, right? So if we have a waxy cuticle, it reduces the evaporation. If we're reducing the evaporation, or the water escaping from the leaves, then we are going to conserve water. I hope that makes sense. Right, we have fewer stomata. Fewer stomata. So again, that will reduce the evaporation. Right? If there's fewer gaps through which we can lose water, that's going to result in more water conservation. Some adaptations are, for example, hairy leaves. So what does that do? Well, hairy leaves, this causes a reduction in air movement. right? So this hairy leaves, around the leaves then, there's a reduction in the air movement, okay? Now, if there's a reduction in air movement, as we've seen, and this connects up to the factors affecting transpiration, a reduction in air movement will result in less evaporation. I think by this point you get what I'm talking about here, so I'm not going to go through each one, but for example, leaf loss. Now, if we lose the leaves, then we kind of reduce the pathway. We just reduce water loss, okay, because we reduce transpiration, okay? Remember that the leaf is the main way through which the plant's going to lose water. You lose the leaves, you know, the rate of transpiration will plummet, and therefore you're going to conserve more water. Right, hydrophytes then. I think this is just a way for you to prove that you understand the principles of this stuff. So again, just a few examples. This is not going to be a thorough list, okay? But just so that you get it, that's the point. So adaptations of hydrophytes. So these plants live in an abundance of water, right? So I guess it's just another way to understand that, you know, what the adaptations of xerophytes are, just to contrast that with plants that live in an abundance of water. For example, hydrophytes have a thin, waxy cuticle. Again, because conservation of water is not really an issue. In fact, we might need to allow more water to evaporate so that we can get that movement of water through the stems, right? So thin waxy cuticle. What else do we have? We have stomata on upper surface of the leaf, right? Again, the movement of water through the plant is not just about water, it's about the mineral ions. Yeah, it's not just about the supply of water, it's also to do with what that water contains. So whether this plant is in a desert or or there's a plant in the ocean, it does still need movement of water through the plants to ensure proper transport from one organ to another. So we need movement in any case. Now, when you have a plant that is surrounded by water, you still need this transport, and so you still need water loss. You still need to have water loss in order to get the movement through the xylem, right? So these plants would have stomata on their upper surface so that they can lose some water through evaporation, so that there's some reason, some driving force for the continued movement of water through the xylem vessels in the stem. Okay, we have small roots. Last one now. Small roots. There are other things, but I'm not going to go through. You get the idea. I think I feel like I'm beating a dead horse here. Okay, we've got small roots because we simply don't need the support. We don't need extensive root systems to try and maximize water gain, because we can rely on water diffusion. Right? Water can reach most of the parts of the plant simply by diffusion because the plant is submerged. Okay? So we don't need that root or the root system to gain that water. Okay? But I think we get it. Right? So this is, I hope that, you know, this has been a story that kind of ties everything together at the least. Okay? If I filled in some conceptual details, then that's great. Right? Again, this is less about what you need to know and more about, you know, the underlying concepts that glues it all together into some kind of cohesive structure. Because if it makes sense, you can remember it. If it makes sense, you can apply it. Okay guys, I'm done. Hope it's been useful. let me know if there's anything about the format or anything that I could optimize or improve in the comments. Thanks guys, good luck.